and welcome to Unit 15.1 of this training module on hot water treatments for seed health. In this unit, we will focus on seed-borne and seed-transmitted pathogens, their definition, distinctions, sources of infection, and their relevance for seed and plant health. We begin with the distinction between seed-borne and seed-transmitted pathogens. Seed-borne pathogens are those carried by seeds either externally or internally. These pathogens may or may not be transmitted to emerging plant. In some cases, the pathogen is present on the seed but does not cause the disease. Seed-transmitted pathogens, in contrast, are directly transferred from seed to the growing plant, where they probably cause the disease. Understanding this distinction is critical as it determines whether a pathogen remains a latent risk or it develops into an active disease problem in crop. A second important distinction is between contamination and infection. Contamination actually refers to the presence of the pathogen on the seed surface. This usually is easier to manage, and treatments like hot water or seed cleaning, like a brushing, can remove this surface inoculum. Infection, however, means that the pathogen has penetrated into the seed tissue. It can be endosperm or the embryo. And in this case, eradication becomes more difficult and the risk of transmission to the plant is much higher. Seed-borne diseases have several significant consequences. They can reduce germination and seed vigor. They can lead to yield reduction in the field. They can lower the quality and the quantity of marketable yield, and they shorten the shelf life of seeds. And this is the reason why seed health management is, let's say, a core of sustainable. Let us briefly review the diversity of organisms that can be a seed-borne. A pathogen, by definition, is a living organism that can cause the disease in plants, in this case, and seed-borne pathogens may include viroids, viruses, phytoplasmas, bacteria, mycota, fungi, nematodes. Each of these groups present distinct challenge for detection and also control. And on the right side, you can see the figure that is representing a plant cell and various pathogens, just illustrating the relative sizes. On this slide, you can see the survival of different groups of pathogens in and on seeds over time. Starting with the fungi on the top, many fungal pathogens survive 5 to 10 years, with around 40-45% still viable in this period. However, their survival drops significantly after the 10 years, and only a very small portion persists above 15 years. For bacteria in the middle graph, the picture is quite different. The majority survives only 5 years. It's more than 65% detected during this period, and few can persist somewhat longer time, 10 or 15 years, but generally they live shorter compared to the fungi. And finally, the viruses in the bottom graph show a very interesting pattern, I would say. Most survive only up to 5 years, but a small fraction remains infection for a very long time, even beyond 20 years. And this is one of the aspects that makes viruses particularly critical for seed health management. They can simply remain, let's say, a hidden reservoir of disease across generations. In a summary, the survival capacity of pathogens differs greatly between fungi, bacteria, and viruses. But all of them demonstrate that seeds can carry viable pathogens for extended periods, and this emphasizes the importance of careful seed health testing. Where do seed-borne pathogens come from? And the sources are diverse, and they include parent plant, soil and crop residues, irrigation water, other crops, weeds, and insects, which can be vectors of pathogens. Pathogens can infect seeds at all stages of development. During the anthesis, the period of flowering, pollen shed, and pollination, pathogens such as smuts or ergots and viruses may infect seeds. During the seed development, systemic infection can establish through the mother plant and carrying the pathogen into developing seeds. And finally, during seed maturation and drying, pathogens may infect or proliferate also post-harvest. More specifically, seed infection can occur through several pathways: the stigma, style, and ovary, as in the case of ergots, loose mats, fusarium, some fusarium species, botrytis, cladosporium, but also, for example, cucumber mosaic virus. Then systemic infection through the mother plant. As I mentioned, they can transmit the pathogen into the embryo, and these are, for example, some embryo-borne viruses, also some of the bacterial diseases, fusarium species, but also vascular wilt fungi or additional, like, for example, verticillium. And then also pathogen can be transmitted via infected pollen, and this is known pathway for some viruses. So to summarize, seedborne pathogens are carried by seed, but not all are transmitted to the plant. Seed transmitted pathogens directly infect the emerging plant and can cause disease. Contamination affects the seed surface, while when we talk about infection, it's about the penetration of the pathogen in the internal tissues of the seeds. Seed-borne diseases can reduce germination, yield, but also shelf life of the product. Pathogens can originate from many different sources and infect seeds at multiple stages of development, and they can also have different pathways. And the next unit, we will look more at the contamination thresholds and detection methods, which can help us in quantifying and managing these risks. Thank you very much.